Welcome to solverfy.
All right, we are being asked to determine which of the following choices is correct regarding DNA sequencing methods and their correct description. So let's start with the first one, the chain termination method. The chain termination method is a method where you want to sequence a certain target DNA, and you put that target DNA in a flask. Let me try and draw a flask here. All right, you put that DNA in a flask along with some nucleotides, right? Nucleotide triphosphates, and along with some dideoxynucleotide triphosphates. Okay, and they go all in there with the DNA. And as the DNA is growing, so let me go over here now. All right, so this is our target DNA. And you put a primer on the DNA and you start replicating the DNA after the primer. Now, as the DNA starts growing, it's going to get incorporated regular nucleotides in there, but sometimes, just by chance alone, since they are in solution, the dideoxynucleotide will get incorporated into the chain. And that can happen either towards the beginning, towards the end, or somewhere in the middle. And the addition of a dideoxynucleotide to the chain terminates replication. It stops replication. Right? Which is why it's called chain termination method. So basically you start producing a bunch of different chains, and just by chance alone, those chains are going to stop reproducing or not are going to stop being replicated at some point in time just by the addition of the dideoxynucleotide, and then you sequence all of those chains, and you can then see that, okay, if the replicated DNA is one nucleotide long and the first ddNTP that was incorporated is a G, then the first letter is a G. And then you say, okay, if the replicated DNA stretch is 2 nucleotides long and the second letter is a T, well, then we know that we have G and T for the first, right? For the first and second. So now that you know the first and second, you pick the fragment with 3 nucleotides long, and that one is also a T. Okay, so we have GTT, and that's how you build the entire sequence. And this is a little bit time consuming, but this is how we first used to determine the sequence of DNA. Now let's move into shotgun sequencing. So shotgun sequencing basically does multiple chain termination methods at once. So instead of just taking one fragment of DNA and making a bunch of different length replications of that and then sequencing each one of those, what you do instead is you take the DNA, you break it into a bunch of different fragments, and then you sequence every single one of those fragments by the incorporation of a ddNTP here. So you basically determine what that last nucleotide was here, and once you have all the different fragments, you can align them by finding sequences of similarity. So you're like, oh, okay, this is an overlap, so that means that the end of this fragment must match to the beginning of that fragment. So you incorporate that into the sequence, and at the end you have the sequence of the large DNA molecule. It's called shotgun sequencing because you basically do a bunch at the same time, not one at a time. And next gen sequencing, I don't have a good graphic for this, but it is basically an automated process that performs thousands of these guys simultaneously. So it's an automated process with like robotic arms and robotic pipettes, and the DNA sequences get determined, and then those sequences are aligned in a computer, and the computer spits out one long DNA sequence. So that's how that works. Now let's look at our answer choices. Choice A, chain termination method is automated sequencing. Well, that is wrong right away, because in chain termination, which was this first guy over here, this is how we used to do it back in the day. There was no automated sequencers here. Choice B, chain termination method. Incorporation of ddNTPs during DNA replication. Yes. Shotgun sequencing. Cutting DNA into random fragments, sequencing using chain termination, and assembling overlapping sequences. Yes, that is correct. Choice, the next thing in choice B is next generation sequencing. Automated sequencers are used to generate sequences of short fragments, and that is correct because it is automated. Choice C is wrong because, well, actually, let me read it to you and then. So choice C, chain termination method, incorporation of ddNTP during DNA replication. Yes, that is correct. Shotgun sequencing, automated sequences are used. No, in shotgun sequences, you don't have automated sequencers. So choice C is wrong. And choice D, that's wrong for the same reason as choice A, because it says that automated sequencers are used in the chain termination method. So the only answer here that has all of the correct descriptions is choice B.